This actually was the first online coverage that we have, and really put us on the map. And then we got covered by several news outlets in China, in the United States, in Europe. So basically, what even when he interviewed us and said realized that we are achieving 1 nanometer or better resolution, and we're starting to see the shape of proteins, and in ancient microscopes, basically that requires really tiny bit fraction of the cost, and these gels, each gel would cost less than €9. So the sample preparation, very cheap chemicals, and your regular light microscope available in your facility would do it. So I said oh wow, you're actually bringing the super resolution to not just for the rich few labs, but to the many, basically, to all labs, not necessarily in the Western rich world, but this can be equally done in the global South, basically. So, and then they came up with the term we're democratizing super resolution. I think I enjoyed the term, but to be fair, there's also a lot of other practices and attempts from different labs that also work in this direction, not just us. And this accessibility of these tools actually opens a lot of avenues for investigators, regardless of their funding thing. And one more aspect that this technology also, let's rethink this up, let's reapproach it. Let's say that you are a super rich lab and you have all the equipments you need and all the personnel you need for that. So then one microscopy might not really be relevant for you. Well, I would like to argue on the contrary, it might still be relevant for a reason where we demonstrated, back to the GABA receptor, is that we know that the GABA receptor, the ground truth structure of the GABA receptor, is missing around 20 to 30% of it, which could not be resolved in cryo-EM, and it could not even be resolved in X-ray crystallography. Why so? Because we know that these domains exist from the sequence, but we never saw them because they are very disorganized. And if you try to average hundreds of thousands to 1 million of these images into reconstructing 3D shape, they average out, and we could not really crystallize them to resolve these structures, while we could see the GABA receptor from one image, one snapshot at one receptor, we could see the last part. And if I may share this with the audience, the first time I saw it, I went to Silvio, I wrote him on Sunday that evening, okay, we see the GABA receptor. It doesn't look nice, actually, because it's longer, way longer than what I see the crystal structure, the PDB structure of what is published. What is happening? Is it not isoform, uniform expansion? Are we really squishing the structure? And we stayed stuck for 2 weeks until we said, okay, it is what it is. Let's speak with our collaborator. Let's see what Rado thinks about it. And when we showed them the images, they're like, you guys, you're crazy. This is so beautiful. I think we're looking at the parts that were missing in this receptor. And it was true. You can also see it that it's predicted to be there in AlphaFold. And the structure that we could eventually using AI neural network that builds on existing solution, CryoFire, that wasn't worlds apart from what AlphaFold predicted there.
Mm-hmm. AlphaFold, by the way, is this very fancy invention that actually people who invented it got a Nobel Prize for it, and it's kind of a algorithm that is predicting shapes of proteins, and there is a database, and you have a lot of proteins there, protein structures, and people who have already solved the structures, they can match it or complement it. And it's actually amazing that you have another tool that you can complementary really find the real truth that is there and in terms of structure of proteins. So you have on one hand accessible and cheap technology that can be used throughout the world, and you do not need expensive microscopes that are worth millions. Then it's faster, way faster than the heavy cryo-EM and X-ray crystallography, for example. And also, it allows you to see things that are not able to be seen with the other technologies.
We can give actually more example on that. So, for instance, there is this M-Cling membrane dye that Professor Rizzoli invented some 15 years ago. It's 7 amino acids, I believe, lysines, with a fluorophore attached to it and a palmitate group. This peptide is very doesn't have density basically. You cannot see it in cryo-EM. And this we could see it basically. We basically labeled the side chains, the amine groups in these lysines with certain chemistry in one color. And then we saw the fluorophore, the head of this peptide in a second color. Basically, so we also can see very small structures that doesn't have enough electron density to be resolved in cryo-EM, but also we can see it in two colors. Like electron microscopy is basically one color. So you can see it in 2 and 3. You can be creative basically there.